everyone, my name is Wilma Richiez-Mateo and I'm currently a third-year human biology student at the University of Puerto Rico in Bayamón. As a Neuro-ID student, I'm conducting research in the medical sciences campus within the lab of Dr. Jennifer Barreto Estrada about addiction. The current exponential growth of opioid overdoses each year has caused a public health crisis that is intervening with the lives of thousands of people. Therefore, I want to do research about the underlying biological mechanism of opioid addiction because it is imperative in order to find appropriate treatments. Consequently, I'm conducting a research project titled BDNF expression in the mesolimbic reward system after extinction of opioid-seeking behavior in female rats. The reason we study the reward circuit is because it is in charge of reward seeking, and in this case, the reward would be a drug, morphine. This system is composed of the nucleus accumbens, the prefrontal cortex, the amygdala, and the hippocampus. BDNF, or brain-derived neurotrophic factor, is a protein in charge of neuroplasticity, and given that addiction is believed to be caused by neuroplasticity damage, we hypothesize that animals that have a higher expression of BDNF will extinguish their morphine-seeking behavior quicker, as well as they will present less withdrawal symptoms. Furthermore, there is a lack of female models being used in the scientific community. Therefore, we wanted to develop our research project with female rats in order to compare if there are any type of underlying biological mechanisms that vary between sexes. To prove our hypothesis, we subject our rats to a conditioned place preference paradigm, or CPP. In this process, we have a two-sided box with different colors and textures, and we place the rat inside. The site the rat spends the least amount of time in will be the site of morphine administration, whereas the other site will be the site of saline solution administration. We will do this because we want the rat to continue to go to a site due to morphine administration, and not just because they like the site because of the color or texture. We administer these solutions 8 days intermittently. After this, we will determine if the rat has extinguished their morphine-seeking behavior. If the rat goes to the site of morphine administration, this means the rat has not extinguished their morphine-seeking behavior because they continuously return to look for the drug. Our preliminary results show so far that female rats and male rats have very similar conditioning patterns, as well as BDNF expressions within the hippocampus. Furthermore, female rats have shown similar accounts for withdrawal symptoms. However, all of these results are preliminary given that we have different sample sizes, but we look forward for doing more research in order to contribute new changes to the public health crisis of opiate addiction.